When to the antibody, antigen is bound. Because of binding of the antigen to the antibody IgG, the Fc region of the antibody will be exposed, which can now bind to the C1 component of the complement system. The first component of C1 component to bind is C1q. When C1q is bound, it will activate C1R, and C1R then activates C1s component. Once the C1s component is activated, it will act as a enzyme, serine protease enzyme. The C1s component will then activate C4 and C2. So because of the action of C1s on C4, C4 will get differentiated into C4a and C4b. And C2 will get differentiated into C2b and C2a. Only C2. C2 is the only exception, where C2a is the large fragment and C2b is the small fragment. Now in the next step, this C4b will combine with C2a. Once they are combined, C4b2a can be also called as C3 convertase. What C3 convertase does is, C3 convertase is the enzyme which can cleave C3. So it will cleave C3 into C3a and C3b. Fine? This is called as C5 convertase. That means it can cleave C5 into two fragments, C5a and C5b. Right? We'll go and attach to C6, C7, C8. Like this. And the membrane attack complex will be formed.